The fourth step of the process of recombinant DNA technology after the amplification that comes is the insertion of the recombinant DNA into the host cell or organism. Now, the recombinant DNA is introduced into the host cell through a process called transformation. That is, transformation can be described as a process in which the recombinant DNA is introduced into the host cell. Now, the recipient cells that gives the DNA after making them competent to receive, that is the host cell, pick up the DNA that is present within their surrounding. Therefore, after this, if a recombinant DNA possesses antibiotic resistant gene, therefore, after transferring into host cell, that is transferring into the E. coli, here we are considering this bacteria as an example, the host cell becomes transformed into ampicillin resistant cell. And therefore, if we put such transformed cells on agar plates containing ampicillin in order to study it or test it, only transformants would grow, untransformed recipient cell will die. That is, in order to check that the process of transformation has been executed or not, the transformed cells, that is the cells on which such transformation process has been executed, are placed on agar plates. that contains the antibiotic ampicillin. Thus, on these plates, only transformants would grow. Or we can say that transformants would live, while the untransformed cells or the cells which did not undergo transformation would die. As a result of this, the ampicillin resistant gene which we have selected as a medium for transformation here is considered as a selectable marker. And so, here in this process of insertion, that is transformation, ampicillin resistant gene can be called as selectable marker. This figure depicts the process of recombinant DNA technology. As it can be seen from the figure, there is a plasmid and a foreign DNA which is inserted into the plasmid. As a result of hybridization, the recombinant DNA is formed, which is then inserted into the bacterial chromosome, which after the process of cloning releases various clones of such recombinant DNA containing bacteria.